Hello students, welcome on board. This is part 5 of the chapter Sexual Reproduction in Flowering Plants. In this session, we will be covering Endosperm Development, Embryo Development, Seeds and Fruits, Apomixis and Polyembryony. For better understanding, please do watch the previous videos. Without further ado, let's begin with the first topic. In the previous videos, we've learned about the processes involved in fertilization, right? Now, in this video, we'll be learning about the changes that take place after fertilization, and these changes include embryo development, endosperm development, maturation of ovules into seeds, and maturation of ovary into fruits. Alright. Now these events together are called post-fertilization events. They are called post-fertilization events. So in this session, we will be covering post-fertilization events and also the structures involved in these processes. Alright. In part 3 of this chapter, we've learned that during fertilization, one of the male gamete fuses with the egg cell to form a zygote, and the second one fuses with the polar nuclei to form the primary endosperm nucleus. It forms primary endosperm nucleus, right? Now here we will be learning how the primary endosperm nucleus develops into an endosperm in order to provide nourishment to the developing embryo. So first, the primary endosperm nucleus undergo repeated mitotic divisions without cytokinesis, which means that there will be no cell wall formation to form free nuclei called nuclear endosperm. Alright. Now, in nuclear endosperm, the cell wall formation begins to form the endosperm, and the endosperm becomes cellular, right? Now, this endosperm then provides nourishment to the developing embryo. All right. So the primary endosperm nucleus first undergo repeated mitotic divisions without cytokinesis to form nuclear endosperm. Then it undergoes cell wall formation to form the endosperm. Alright. Now, in some plants, the endosperm is completely utilized before maturation, and these are called non-albuminous seeds. And in some other plants, a portion of endosperm remain even after maturation, and these are called albuminous seeds. Alright. Now we'll learn about these seeds in detail shortly. Alright, so we've learned about the endosperm development. Now we're going to learn about the embryo development. Okay? The embryo develops at the micropylar end. It develops at the micropylar end of the embryo sac, where the zygote is situated. Alright? Now the zygote begins to divide only when certain amount of endosperm is formed. This is because the endosperm provides nourishment to the developing embryo. So the embryo develops at the micropylar end where the zygote is situated, and the zygote begins to divide only when certain amount of endosperm is formed in order to derive nutrition from it. Alright, now the early stages of embryonic development, embryonic development are similar in both monocots and dicots. Now, as you can see in this picture, the zygote give rise to a proembryo, which give rise to a globular embryo, then to a heart-shaped embryo, and finally to a mature embryo. So, these stages are common in both monocots and dicots, and these are the early stages of embryonic development. All right. Now, we learn about dicot embryo. Dicot embryo. In this picture, you can see that dicot embryo has two cotyledons. These are the cotyledons, and it has an embryonic axis at the center. Alright, now epicotyle is the upper part of the embryo and hypocotyle is the lower part of the embryo. Epicotyle is basically the portion of embryonal axis above the level of cotyledons, and it terminates to form plumule or stem tip. Alright, and the cylindrical portion below the level of cotyledon is called hypocotyle, which terminates to form the radicle or root tip. And the root tip is covered with a root cap. Alright, so now we've learned that a dicot embryo has two cotyledons, and at the center there is an embryonal axis. Alright. The upper region is called epicotyle, which is the portion of embryonal axis, above the level of cotyledons, which terminates with the plumule or stem tip. Alright. And the lower part is called the hypocotyle, which is a cylindrical portion, below the level of cotyledons, that terminates to form radicle or the root tip. And this root tip is covered with a root cap. Alright. Now in a monocot embryo, there is only a single cotyledon. Which is called the scutellum. Now, in a monocot embryo, at the lower end or at the hypocotyle, the embryonal axis has a radicle. It has a radicle and a root cap that is covered by an undifferentiated sheath called coleorhiza. It's called coleorhiza. So, at the hypocotyle or at the lower end, the embryonal axis has a radicle cap, a root cap, which is covered with the undifferentiated sheath called coleorhiza. And at the upper end or at the epicotyle, the embryonal axis has a plumule or a shoot apex with few leaf primordia, and it is covered with a hollow foliar sheath that is called coleoptile. It's called coleoptile. Alright, so we've learned that at the lower end or at the hypocotyle, the embryonal axis has a radicle and a root cap that is covered by an undifferentiated sheath called coleorhiza. And at the upper end or at the epicotyle, the embryonal axis has a plumule, or a shoot apex that is covered with a hollow foliar sheath called coleoptile. All right. Now we learn about seeds. A fertilized ovule is called a seed. A fertilized ovule is called a seed. And it is the final product of sexual reproduction. So a fertilized ovule is called a seed, and it is the final product of sexual reproduction. Now a seed consists of 3 parts: a seed coat, cotyledons, and an embryo axis. So it has 3 parts: a seed coat, cotyledons, and embryo axis. First, we learn about the seed coat. All right. Now, in the previous videos, we've learned that the ovules are covered with 2 integuments, an outer integument and an inner integument. Now, these integuments later harden to form a tough and protective seed coat around the seed. So the integuments around the ovule harden to form a tough and protective seed coat. All right. And the micropyle remains as a small pore. In the seed that facilitates the entry of oxygen and water into the seed during germination. Now, micropyle is the region where the integuments are absent, and this micropyle remains as a small pore in the seed which facilitates the entry of oxygen and water during germination. All right. Now, as the seeds mature, their water content decreases, and therefore the seeds become dry. And at this stage, the metabolic activity is reduced, and the embryo enters a state of inactivity. Which is called dormancy. So as the seeds mature, the water content is reduced and the seeds become dry. And at this stage, the metabolic activity is reduced and the embryo enters a state of inactivity, which is called dormancy. Now, when favorable conditions are available, the seeds begin to germinate. Now, favorable conditions include adequate water, oxygen, temperature, etc. All right, so we've learned about seed coat. Now we learn about cotyledons. Cotyledons are basically simple structures which are generally thick and swollen as they contain food reserves. All right, So cotyledons are simple structures which are generally thick and swollen as they contain food reserves. So that is about cotyledon. Now we learn about two types of mature seeds. The first one is non-albuminous seeds. Non-albuminous seeds. Non-albuminous seeds or ex-albuminous seeds or ex-albuminous seeds. Alright. So non-albuminous seeds or ex-albuminous seeds, they have no residual endosperm, as the endosperm is completely consumed, as it is completely consumed during the embryo development. Alright. So non-albuminous seeds, they have no residual endosperm as it is completely consumed during embryo development. Now the examples of non-albuminous seeds include pea and groundnut. Pea and groundnut. Alright. The next one is albuminous seeds. Albuminous seeds. Now albuminous seeds, they retain a part of endosperm, as the endosperm is not completely utilized during embryo development. Alright. So albuminous seeds retain a part of endosperm as it is not completely utilized during embryo development. An example of albuminous seeds include wheat, maize, castor, etc. Now in some seeds, the remnants of nucellus are persistent and it is called perisperm. Alright, so we've learned about the different parts of the seed and we've also learned about two types of seeds, right? Now here we'll be learning the advantages of seeds. The first one is that the seeds help the species. They help the species to spread in different areas, or other areas through dispersal. Alright? They help to generate new combinations leading to variation among the species. Seeds are also stored and used as food throughout the year, and they can be used in favorable seasons. Depending on their viability. Alright. So the advantages are seeds help species to spread in other areas through dispersal. They generate new combinations leading to variation in the species. Seeds are stored and used as food throughout the year, and they can be used in favorable seasons depending on their viability. Alright. Now we learn about fruits and different types of fruits. Alright. So we've already learned that seeds develop from ovules. Similarly, fruits, they develop from ovary. So ovary develop into fruits. So that means wall of the ovary develop into wall of the fruit, and it is called pericarp. Alright. So wall of the ovary develops into wall of the fruit, which is called pericarp. Now the fruits that develop from ovary, they are called true fruits. True fruits are the fruits that develop from the ovary. Alright. Now the examples include mango, orange, etc. Now in some plants, when the ovary develops into the fruit, other floral parts degenerate or fall off. Alright. Now the second type of fruits are called false fruits. These are fruits that develop from the thalamus. They develop from the thalamus. Examples include cashew, apple, etc. By now we know that the fruits are result of fertilization, right? Now there are some fruits that develop without fertilization, and these fruits are called parthenocarpic fruits. Parthenocarpic fruits. Parthenocarpic fruits are the fruits that develop without fertilization. They are the fruits that develop without fertilization. An example is banana. Alright. Now parthenocarpy can be induced using growth hormones, and such fruits will be seedless. Alright. So we've learned about 3 fruits, right? The first one is true fruits, which are fruits that develop from the ovary. The second one, false fruit. They are the fruits that develop from the thalamus of the flower. And parthenocarpic fruits are the fruits that develop without fertilization. Alright, so now we've reached the last topics of this chapter, which is apomixis and polyembryony. We'll begin with apomixis. So apomixis is a special mechanism. It's a special mechanism that produces seeds without fertilization. It is a special mechanism that produces seeds without fertilization. It is a form of asexual reproduction. That mimics sexual reproduction. Alright. An example for this is grass. Alright, so apomixis is a special mechanism in which seeds are produced without fertilization. It is a form of asexual reproduction that mimics sexual reproduction. An example is grass. Alright, now moving on to polyembryony. Polyembryony is the presence of more than one embryo in a seed. Polyembryony is the presence of more than one embryo in a seed. Alright. So that's it for the session. This was part 5, where we've learned about endosperm development, embryo development, seeds and fruits, apomixis and polyembryony. With this, we come to the end of the chapter, sexual reproduction in flowering plants. If you found this helpful, please do like the video, and if you have any doubts, comment down below. Subscribe to watch the upcoming videos. Thank you for watching.